Hello, thank you for joining me here today at the Soil Water Conservation Society virtual meeting. My name is Shannon Cappellazzi. I am the Director of Research at Grassland, Oregon, where we breed improved varieties of cover crops as well as perennials and grasses. I'm excited to share my research today with you with a general overarching question about whether cover crops can break through restrictive soil layers. And we have a kind of interesting setup for how we will do this research. It's what I lovingly refer to as our tubes in the ground project. And the impetus for using the tubes in the ground in this way came from a question from a farmer, which is where I think all the best questions, they come from. So we were at a growers meeting out in Iowa, and there was a group of many progressive farmers. A lot of them had been no-tilling for 30, 50 years and cover cropping for 10 to 30. And this one producer came up to me after we had our meeting, and he said, I've got this problem and I don't know what's going on. He's like, I've been doing no-till for 30 years. For the most part, things are good. I use anhydrous for injection on my nitrogen. The corn started out great this year, and then it just petered out. And I asked him, if he looked under the soil. Right? I'm working at a plant breeding company, but they hired a soil scientist to be their director of research because they wanted everyone to start flipping that idea, thinking about what's going on in the below ground components of the plant and what that means for the above ground health. And he got really excited. He had dug up the plant. He went out to his truck and he grabbed a couple of them out of the back of his truck. And you could just see that they had this really weak root system. And not only was it weak, but it went down a couple of inches, maybe 4, 6 inches, something like that. And then it just went sideways. And I would call that J hook. It looks as if the root is progressing and then it just feels something that it doesn't like and tries to go to a different area. So it was going laterally instead of to continue to go down. And so his question was, are there any cover crops that I can use to fix this problem? And we hadn't completely identified what the problem was, but he did have an idea that it could have to do with low soil pH. He had taken measurements, for most of his time where he took a full 12-inch measurement. But he did have somebody who recommended that he take small incremental measurements for pH. He took a measurement every 2 inches for soil pH. And when he did that, that's where he could see that there was some layers with really low soil pH. And so having these conversations, other ones I've had in the past with producers out in Eastern Oregon who are starting to see some really low soil pHs, it kind of led us to this idea about what can we do to evaluate whether or not some of these cover crops that we've been working with have the ability to break through these restrictive layers. So we have this tubes in the ground project that we set up where we were able to have a controlled soil setting. These are 4-inch PVC pipes that you see with, they go 4 feet down into the ground. And then there's these clear plastic sleeves, and those clear plastic sleeves can be removed. And I created an artificial soil horizon that had very specific properties. And so the first one of these restrictive soil layers that we built was with compaction. We know that soil compaction is a common problem. It can often be caused by a tillage implement hitting that same depth over and over again. Maybe it's at 6 inches, maybe it's at 9 inches. And you can start to get these areas with really high bulk density that make it hard for the roots to get through. Typically, when we see a bulk density over 1.6, we start considering that root restrictive. And so what I did is I measured a certain amount of soil, a mass of soil, and I made it fit into the volume that I wanted, so I could try to get to the bulk density that I was aiming for to make these root restrictive layers. So at the top, it was a very low, light organic, lots of organic matter, light bulk density soil. We got a little bit more packed, and then in this third horizon, we get to a bulk density. We were trying to pack it to a bulk density of 1.6. The horizon below that, we didn't pack it as hard specifically, but because we had been packing the horizons above that, that one was at about 1.5, and then below that in the bottom of the tube, we're down to about 1.3 again, a very typical bulk density measurement. The next soil setting, oh, and this is what it looked like as I was packing those tubes. So these are two different tubes. This is one that I had already tamped down, and this is one I had not tamped down yet. So you can see I wetted up the soils kind of periodically as I was adding the soil to it, and then I would tamp it to the right volume. So I had a known amount of soil that I added and tried to hit a specific volume, and you can see that difference in volume between those two soils before and after I had tamped it. The next setting is with the restrictive layer is a low pH. And so one of the things that we've seen in many different fields now is this problem with these small horizons of soil acidification. So we're getting this horizonization where, particularly in situations where people are using no-till and synthetic nitrogen, somewhere between 4 to 9 inches below the surface, depending on their practices, they're getting this zone that has really low soil pH, and roots don't want to go through it, particularly not the corn and the wheat roots that we've sampled in the past. We have soils over here in Eastern Oregon that typically have a pH around 6.8, and they're measuring all the way down to 4.2. So I did a titration in the laboratory to determine how much elemental sulfur I would need in order to lower the pH of these soils. And then as I was building each of these soil profiles, I added a certain amount of dry elemental sulfur to the soil as I was building them for each one of these layers. And then we had also a soil setting that I just considered a control. This should have no root restrictive features. It's a sandy loam mix with compost with bulk density around 1 and a pH around 6.8. I didn't add any fertilizer, but I didn't think that this was going to have any kind of restrictions for the root growth. Sorry, that keeps advancing double. There were 6 different treatments that we considered these cover crop treatments. And so this first one that I have abbreviated ARG is annual ryegrass. It is a Lone Star variety. The next abbreviation is IAR, and that one we have inoculated with our buscular mycorrhizal fungi with that same annual ryegrass, the Lone Star annual ryegrass. So ARG and IAR are 2 ryegrasses. RGB is annual ryegrass with Balansa fixation clover. And then we have one I abbreviated TRT for a tap-rooted turnip. This is an experimental variety we are working on right now. And then BCL, you'll see, is the fixation Balansa clover. And CTL, this is the control with no plant. So in each one of the soil settings, I also have a control where there is no plant growing. And then, we planted everything around the middle of September last year, planted everything at the same time. After we randomized everything out in the field, we had 4 replications of each of the treatments for 3 different destructive sampling dates. So, there's 216 of those tubes out in the ground. And then our first destructive sampling time was at the time that we would terminate the cover crops. And so, around the beginning of June, we were terminating these cover crops. And before that, we had measured standing water, we had measured plant height, we took the above ground biomass measurements. And then once we got into the soil, we took the tubes that you can see here, we laid them on this little contraption that we made with guides for our saw. And we literally sawed these tubes into these smaller groups so that we could make sure that we had actually hit the bulk density that we were trying to hit, that we could sample the pH by horizon, and that we could assess the root mass by horizon. So you can see here, we had a root washing system. We took out the roots and weighed them all by horizon. We have all of those roots saved so that we can do further analysis if we need down the road. And that's kind of that first step. So we're measuring the soil and the roots by horizon for each of these soil settings. The first thing that we noticed is that the control soil setting did not perform very well. This is the control soil here, and you can see this is root biomass all in horizon 1 right at the top before there should be any restrictions. And the control soil, there was just very low root biomass. You can see this at the above ground biomass as well. The high bulk density soil and the low pH soil, those ones were considerably higher than the control, which I thought would be non-restrictive. Before I pull up the figure here, I know that there's way too much data, but I do want to draw your attention to a couple of things. So bear with me here with this big busy figure. So we've got this control soil, and the first thing, just to draw your attention, that control soil, no matter what depth we are, no matter what plant we are, it didn't do very well. Okay, so we're going to kind of set that aside. And then another thing you might notice is that the tap-rooted turnip, that TRT treatment, had really low root biomass. And there's a couple things going on here. It's a very different root type than these others. But when we were first pulling these tubes out of the ground and checking them periodically, it was the first one to send roots down all the way to the bottom of the tube, and it looked great. Everything was happy. When we terminated them, those roots were still down there and happy, but we did terminate that taprooted turnip about a month and a half before we terminated the other cover crop types. So there could have been some decomposition of that root biomass by the time we got to actually taking these measurements. The next thing that I want to point out is that in this high bulk density soil, each of these are horizons, right? So this top one is horizon 1, and then we have horizon 2, horizon 3. And what I wanted to see is that it looks as if there's a pretty big drop off in the root biomass between horizon 3 and horizon 4 here in the high bulk density soil. And then in the low pH soil, it looks like there's a relatively big drop off between horizon 2 and horizon 3. And we'll see that in the data as we go through a little bit more. This is the slide that I am the most excited about. And to explain what is going on here, this is a visual representation of what's called a Dunnett's test for multiple comparisons to a control. So what this figure is showing is along this middle line here, this is the average of the controls for all of the soils that had a low pH. And so the control here is the low pH soil setting that had no plant in it. And so the pH was about 4.1. You'll notice that there are two treatments that have these red dots and are outside this upper boundary, and that means that they're statistically higher than the control. And we'll notice on this one that these are both ryegrass treatments. And this other one that's kind of tending towards, it's getting close to that line, that's also ryegrass. And when we move over to horizon 3, we do have a lower pH again here. Our control, the mean was all the way down to 3.4, which means I need to work on my titration for the next one. Hopefully I'll get that a little bit more dialed in and not drop the pH quite so low. But our control pH was at 3.4. And again, there are 3 treatments here that are all statistically higher than the control. And all 3 of them are the only 3 treatments in here that contain ryegrass. And then again, in horizon 5, we have a very low pH, 3.7. The 3 treatments that had higher pH, and at each of them, that's about a point higher. So 3.7 to 4.7. It's those treatments that have ryegrass in them. So that's something that we're going to follow up on in our next year's worth of trials. When it comes to the bulk density, one of the things that we had noticed early on was that there was standing water in a lot of our bulk density soil settings. There was not standing water in the low pH or the control setting. So that, kind of gave us some confidence that we're doing a good job of getting that really packed in there tight. When we look at that horizon 2, before we had done any compaction, the mean for the soil bulk density was at 1.8, and there was no significant difference among any of the treatments at that high level. But then when we get to that next horizon, that horizon 3, and you'll remember there were plenty of roots still in horizon 3 in that bulk density. The mean for the control was at 1.56, so we got pretty close to what we were trying to hit with that 1.6 bulk density. But in every single one of the treatments that has plants, there's a much lower bulk density, and it's considerably lower. It's down to 1.25, which is a very healthy level for roots to be able to get to. Then we get to horizon 4, where the control was at 1.4. It was set around where we thought we would be able to get it. Most of the treatments are not different than that. The inoculated annual ryegrass is a little bit higher, and they do tend towards being a little bit higher. But there's something going on here with horizon 3. Those roots have pushed through at least that deep. Maybe they've moved some of the soil down deeper. We're not quite sure. There could be some artifacts from the way that we set up the experiment. So we will evaluate our methods and try something maybe a little bit different to see if we can replicate it under slightly different conditions. But it almost looks as if there's an influence from the plants on having an influence on that bulk density. So the next steps is, okay, well, we've got some of this information about what's going on with the soil and the roots of the cover crop, but is that going to then influence the success of the subsequent crop? So particularly with the corn. And so we planted, after we terminated all of the cover crops, we planted corn into them. And remember, this is destructive sampling. So I took 4 replicates of each one of the treatments, and I processed all of the, well, our team processed all of those. And then we planted corn into the tubes that were still in the ground. And so here you'll see that we've been kind of checking on them occasionally, and you'll see a lot of roots on that corn, you know, particularly along the outside of the core. And not as much on the inside. So we need to kind of evaluate that if anybody has ideas on it. But we will be measuring corn yield and a greenness index, as well as total root biomass and root biomass by horizon of the corn to see if any of these cover crop treatments had an influence on the success of the corn. And then we'll also have some measurements on differences in soil health. So we'll assess the total carbon and nitrogen, soil organic carbon and nitrogen, potential carbon mineralization rates, as well as aggregate stability. And all of those are by horizon. So we'll do all of those tests for the 72 tubes for the final destructive sampling date with 5 horizons each. So I hope you will be intrigued at least enough to start following the research. Like I was telling you earlier that the taprooted turnips showed this really beautiful root structure down deep into the bottom of the tube earlier in the trial. You know, you can see these orange, some of the pictures you can see really clear glade, and you know, the redox reactions are going on right around the zone of the root. I just think are absolutely beautiful. And this trial gives us the opportunity to see these roots moving through the soil in a way that is pretty unique. So I hope you will follow the research, and you will start to think about ways that we can evaluate these crops to try to overcome production challenges that the producers are working with. And feel free to give me a call or email me anytime. My email is right here. And I want to thank everybody here at Go Seed. We've got an amazing team, and I hope to see you live in person next year.